Hello, 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 hello. Am I audible? Hello, very good evening to everyone. Welcome back. Welcome to Goscore Learning, GCC's fastest edtech. Now, again, I am here with the fifth chapter, that is molecular basis of inheritance. This is the last portion of our chapter. So, today we will be discussing on translation. What is the machinery for translation? What are the different steps for translation? And also, we'll be discussing with operon system. Okay, lac operon. What is the speciality of that operon? Then finally, we will be into the Human Genome Project and Rice Genome Project. So, we'll be concluding the chapter today. From this chapter, you know that more weightage will be coming from this particular chapter, and you have to be thorough from the previous portions too. And once again, it is very important chapter. Again, we can look into the topic. We'll be moving into the topic. Okay, so last class we have discussed on the translation missionaries. At that time, we have seen for translation what is there, mRNA should be there, and it has the codon. The triplet is known as codons, right? Each triplet is known as codon. It will code for one particular amino acid. For example, we can say AUG. AUG is a start codon, right? AUG. AUG is a start codon. What is AUG? AUG is a start codon. See, you can find AUG here in this. And what does this AUG codes for? What does this AUG codes for? See, AUG. You can find AUG here, right? AUG here. This is the table 6.1 which is present in your textbook. So you have to take the textbook and just mark what is AUG there. What is AUG? AUG can code for methionine, right? AUG can code for methionine. So different codons codes for different amino acid. Different codons can code for different amino acids. So next, if you are taking an example, you can take UAU. Okay, what is that? UAU. UAU can code for an amino acid known as tryptophan. Okay, what is that? Tryptophan. It can code for an amino acid known as tryptophan. Okay, so you understand, right? Simple it is. Triplets are there. Triplets are acting as codons, and this codons can code for amino acid. One particular codon can code for one particular amino acid. One amino acid can be coded by many different codons. Okay, so last class, we have seen with a features of genetic codon, salient features of codon that we have already discussed. Actually, I told you that there is one particular amino acid which acts as the start codon. Okay, so that start codon is AUG, and amino acid codes for AUG is methionine. Always in translation, methionine will be acting as the initiator amino acid. Okay, so if you are looking into the termination, stop codons will be there. There are three different stop codons for we can say for amino acids. Three different amino acids will no amino acid will be codes for this particular terminator sequences. Okay, so last class we have already discussed the salient features of genetic codon. So today we'll be discussing again with the requirements for translation process. What are the different requirements for translation process? Okay, so first I have told you that it is a long mRNA. Okay, what it should be? It should be the mRNA. It should be the mRNA. Okay, so we'll write it in small letter. Okay, what it is? mRNA should be present for the translation. So after transcription process, right? After transcription process, okay, after transcription process, see transcription, it has taken place in the case of eukaryotes in nucleus. Okay, in the case of eukaryotes, it takes place in nucleus. From there, the mRNA, if you are considering this as nucleus have inside the nucleus what is present? mRNA is present and there is small pores over the surface of nucleus. That is known as nuclear pore. Okay, through this nuclear pore, what will be coming out? mRNA will be coming out into the cytoplasm. So, we can consider this as the cytoplasm. This is in the case of eukaryotes. Okay, so before coming into the cytoplasm, three different steps have already done, that splicing, then we can say capping and tailing. All these processes have done and now the mRNA is in the cytoplasm with different exons. Okay, so next what is the process? This mRNA has to undergo what process? The process is known as translation. Okay, why this process? Why translation? If translation happens, translation can lead to the process or formation of different proteins. Okay, translation helps in the synthesis of proteins. For translation machinery or for translation, now in the cytoplasm, what is there? mRNA is there. mRNA can't translate itself. Okay, it needs different help. What are the requiring material? First, you have said that it is mRNA. Then we have discussed about the genetic codons, right? Genetic codons and amino acid. So, amino acids are important. Okay, what is important? Amino acids are important. Amino acid should be there. So, where does this amino acid is present? This amino acid is present inside the cytoplasm as amino acid pool. Okay, we can say amino acid is present in the cytoplasm as amino acid pool. So, inside this amino acid pool, there are many different types of amino acid. How many amino acids we have studied? 21 amino acids, right? So, this 21 amino acid will be present in the amino acid pool inside the cytoplasm. Okay, so the second requirement is ready. So, the mRNA is present here. Amino acid is present in the amino acid pool. This amino acid has to reach into the site of mRNA. Okay, who will take this amino acid into mRNA? That is next question. Okay, so that third person is tRNA. Okay, what it is? tRNA, or we will call it as transfer RNA. Okay, what RNA it is? Transfer RNA. What is mRNA? What is mRNA? mRNA is just messenger RNA. mRNA is messenger RNA. We'll call tRNA as transfer RNA. Why it is known as transfer RNA? See, amino acid pool, inside amino acid pool, amino acid is present, right? This amino acid has to be taken from the amino acid pool towards the mRNA. Okay, got the concept? See, this amino acid, from amino acid, there are different amino acid like lysine, leucine, isoleucine, then tryptophan, methionine. This is amino acid pool. Okay, inside this many amino acid are present. This amino acid has to be taken from amino acid pool and it has to be transported to mRNA. Okay, it has to reach into mRNA. For that purpose, who is taking this amino acid? tRNA is taking the amino acid. Who is taking tRNA? We will call it as transfer RNA. Okay, so you have mRNA, you have amino acid, then you have tRNA. But for the process of synthesis of protein, you need another important thing. Okay, another important thing which you have studied as one cell organelle. Which is that? Which is that? This is first one, this is second one, this is third one, and next it is very important thing as ribosomes. Okay, ribosomes. Inside the ribosomes, you have studied it is made up of proteins and mRNAs, right? So, sorry, rRNAs. We'll call it as also rRNA. What is rRNA? Ribosomal RNA. Okay, so ribosomes are present. These ribosomes are for the translation purpose. Ribosomes are for translation purpose. You have already studied the structure of ribosome in grade 11 in cell chapter. So, you have to understand what is ribosome. Okay, or what is the structure of ribosome? We'll discuss each and everything in an orderly manner. So you got it right. What is translation? It is the protein synthesis. How the protein is synthesized. This is in the case of eukaryotes. If you are considering prokaryotic translation, both transcription and translation will be taking place simultaneously. Okay, so we can say in the case of the translation, what are the requirements? It is mRNA, amino acid, transfer RNA. Then the last one it is ribosomal RNA or ribosomes. Okay, let's see the structure of tRNA or the transfer RNA. Okay, let's see the structure of transfer RNA. So, if you are considering the structure of transfer RNA, it is like clover leaf structure. What leaf it is? Clover leaf structure. Okay, clover leaf structure. Okay, so if you are drawing the structure, the structure of tRNA is like this. Okay, the structure of tRNA is like this. That means it has a 5 prime end. Okay, it has a 5 prime end. It has a 3 prime end. And it has an anticodon loop. Okay, it has an anticodon loop. We will call this site as anticodon loop. This is a loop structure, right? So we'll call it as anticodon loop. Then this site is DHUR. What this is known as? It is known as DHUR. And this is known as T-psi-C-R. What this arm is known as? It is known as T-psi-C-R. So here, what happens if an mRNA is present like this? Okay, when mRNA is present like this, so we can write A, U, G, U, G, A. Okay, this is the codon. For this particular mRNA, we can say AUG. This is a starting codon, start codon. Okay, for this start codon, AUG, what must be the anticodon? Adenine. Adenine will code for uracil. Okay, uracil will code for adenine. Guanine is here. Guanine will code for cytosine. Okay, so this will be the anticodon structure or anticodon base pair will be acting as UAC, and the 3' end. Here you can find the 3' end. Somewhere amino acid has to be attached, right? So amino acid will be attached to which side? 3' end. We can say here amino acid will be attached. Okay, got it? It is like a clover leaf structure. It has a 5' end. It has a 3' end. Towards the 3' what will be attached? Amino acid will be attached. Okay, and it has two arms known as DHU arm and T-psi-C arm. Okay, here you can find a modified base pairs. Okay, we have different nitrogen base pairs, right? Here in this particular bases, you can find modified base pairs. Okay, then in DHUR you can find dihydrouridine. You can find dihydrouridine as the base pairs. Okay, in THU arm you can find in DHU loop you can find dihydrouridine, and in T-psi-C loop you can find pseudouridine. Okay, here you can find pseudouridine as the amino acid. Okay, and also here you can find ribothiamidine. Study the Base pairs. Okay, what is the name for the base pairs? Okay, so what is present? See, once again, it has a 5 prime end. It has the two different arms. It has an anticodon loop. Okay, and what is the speciality of 3 prime end? It has amino acid end. It is known as amino acid attachment site. Okay, then in the DHU arm and in T-C arm, what is DHU arm? It has the dihydrouridine. In T-C arm, it is also present with pseudouridine or ribothymidine. Okay, so that is the structure of tRNA. We'll look into the structure of tRNA. See, this is also known as adapter molecule. tRNA is also known as adapter molecule. Why it is also known as adapter molecule? It is taking amino acid from amino acid pool and it is connecting to the mRNA. So see, tRNA is a molecule that has about 60% of its parts double stranded and the rest remains single stranded which has unpaired base pairs. Okay, so if you are looking into the structure, see, it has a DHU arm. See, we can say this is the DHU arm. Okay, so this is the DHUR, this is the T side CR, this is the anticodon region, this is the 5 prime end, this is the 3 prime end where the amino acid has connected. Okay, understood the structure. So we'll be moving into the description. I told you that there is an anticodon loop present and it will be the complementary to the codon which gets attached in mRNA. If an mRNA is present, if there is codon, okay, AUG, the anticodon will be UAC. Okay, so that is anticodon. Then amino acid has acceptor end to which amino acid binds and it is at 3' end. It has CCAN. What end it has? It has a CCAN. So if you are looking into 3' end, if you are looking into 3' end, see this is the 3' end, right? So here you have CCAN. What it has? It has CCAN. Okay. Next, if you are looking T-C loop. I told you that there is a T-C loop, but what is the speciality of the T-C loop? It has some unusual base pairs. It has some unusual base pairs, and what is the speciality of this unusual? What are the different unusual amino acid? It is pseudouridine and ribothymidine. You have to remember the names of extra base pairs. Okay, they are unusual or modified base pairs present in T-C loop. Okay, so next one it is DHU loop. What is next? It is DHU loop. What is the speciality? I have told you that there you can find dihydrouridine. Okay, dihydrouridine. So what is the speciality of this dihydrouridine? This dihydrouridine helps, it is the binding site for the enzyme. One particular enzyme is present for the activation of amino acid. Okay, so for the activation of amino acid, and that enzyme is known as amino acyl synthetase. What the enzyme is known as? Amino acyl synthetase. It is important for the activation of amino acid, which we are going to study in the steps of translation. Okay, so there is a small extra arm. I forget to say about this extra arm. So see, extra arm is present in between the T-C loop and anticodon loop. You have to study the location of extra arm. Where does this extra arm is present? I have drawn it in the picture. See, if you are looking into this T-C arm, this is T-C arm, right? This is T-C arm. So we can say in between T-C arm and in between anticodon, there is an extra loop. Okay, there is an extra loop. And that extra loop is known as extra arm. Okay, so I believe that you get the structure of tRNA, the adapter molecule. Okay, so you have now the amino acid, different codons, and you have tRNA for the synthesis. Next, we want to study about the ribosomes, right? Ribosomes, the structure of ribosomes or the different ribosomes. Okay, so we can say what are the different ribosomes? We consider prokaryotes. Okay, what are the organisms? How can organisms be classified? It is prokaryotes, and as eukaryotes. Okay, prokaryotes and eukaryotes. If you are considering the structure of ribosome, you have to understand that it has a larger subunit and also it has a smaller subunit. Okay, it has a larger subunit and also it has a smaller subunit. Okay, understood? Clear. So for if you are considering prokaryotes, the prokaryotes have the subunits as 50s subunit and 30s subunit. Okay, 50s subunit and 30s subunit. The subunits of prokaryotes, ribosomal subunits will be, the larger subunit will be 50s. Okay, the smaller subunit will be 30s. Okay, so next if you are considering, if you are considering the eukaryotic ribosome, the same structure, that means a larger subunit will be there and a smaller subunit will be there. Okay, so if you are looking into the larger and smaller subunit of eukaryotes, it is 60s subunit and it is 40s subunit. Okay, so the whole together the structure contains, we can say they are ribosomes are 70s. Okay, they are 70s, and if you are considering eukaryote, they have 80s. What is this S? S is known to be Sedberg unit. Okay, it is known as the sedimentation coefficient. We will call it as sedimentation coefficient, or we can say that it is Sedberg unit. Okay, so that is the structure of ribosome. And dear students, you have to understand what are the different sites in this ribosomes. Okay, so I told you that is there is a larger subunit. Okay, that there is a larger subunit. My subunit structure is like something, right? So we'll draw it again. Okay, there is a larger subunit and also there is a smaller subunit. Okay, there is a larger subunit and there is a smaller subunit. If you are looking into this particular structure, we have to understand the process of translation will be taking place inside three different sites of ribosomes. Okay, the process taking place inside the three different sites of the ribosomes. Let's see what are the different sites of ribosomes. We can say E site is there. Okay, what is that site known as? E site. Then you can find P site. Then you can find A site. Okay, what are the different site? Okay, what are the different site? We can say that it is E site, P site and A site. Okay, so what is the speciality of E site, P site and A site? Can you comment in the comment box? Can you comment in the comment box? E site, P site and A site. Okay, so if you are considering E site, P site and A site, E site is for, okay, so what is E site and P site and A site? E site is, we will call E site as exit site. Okay, E site as exit site. Then P site as peptidyl tRNA binding site. That means simply peptide bond is forming here. Okay, peptidyl TIP. Oh, okay. It is TI. Peptidyl tRNA binding site. Okay, peptidyl tRNA binding site. And later A site. A site is also known as amino acyl tRNA binding site. Amino acyl tRNA binding site. Okay, got it. So this is the structure of Ribosomes. Okay, ribosome has three different sides: E site, P site, and A site. E site is known as exit site, P site is known as peptidyl tRNA binding site, and A site is amino acyl tRNA binding site. What happens here actually? P site, or we can first consider P site. Okay, what is the function of P site? During translation, the first amino acid is methionine, right? First amino acid, first codon is AUG. Initiator codon is AUG. So, during initiation step, first step is initiation. What is the first step during translation? We can just go through the steps of translation. Okay, steps of translation. The first step is initiation. Initiation. What happens in initiation? In every initiation phase, what will be the amino acid present? What will be the amino acid present? The amino acid present is AUG. Okay, amino acid present is AUG. AUG. And it codes for which amino acid? Methionine. Okay, that is initiation. First step is initiation. Second step is elongation. Okay, third step is termination. Termination. This we have to study. Okay, so in initiation, elongation, and termination, what happens? Okay, in the initiation process, what happens? This amino acid, okay, AUG is the amino acid. AUG is the amino acid. This AUG will come and bind to the which side? P side. Okay, it will come and bind to P side. Okay, so what is second step? Elongation. First amino acid is here now. Second amino acid will come to which side? A side. Okay, after coming the second amino acid, this we can consider second amino acid will be here. First amino acid will be here. Then what happens? This first amino acid will be moving to the second amino acid. Okay, now there is no tRNA with no amino acid. So we can say next amino acid, this two amino acids, 2 and 1 amino acid will be moving to the, again it will move to P side, and again it will move to E side. E side means exit side. Exit side, what is the meaning of exit side? Exit side, the tRNA which is present in P side will be moving to E side. Okay, tRNA which was present in the P side will be moving to E side. From E side, it will be moving out of the ribosomal structure. Okay, so we can say these are the steps. We'll see in detail what is initiation, what is elongation, and what is termination. Just now study what is the structure of ribosome and what are the different sites in what are the different sites in sites present in ribosomes. Got it? So just study what are the different sites. E site, first site E site, then second P and A. Okay. So let's move on to the steps which is involved in, or what are the different steps for translation. I told you there are three different steps. Along with these three steps, an initiative step is there. Okay. You have to understand that if you are considering this as the cell cytoplasm, there is amino acid pool, right? There is amino acid pool here. There is a, okay. There is, we can consider nucleus, a small nucleus is present here, and this is the cytoplasm, and mRNA is now present here in the cytoplasm. Okay, tRNA is also present in the cytoplasm, this tRNA. Okay, tRNA is present in amino acid. Everything is present in, cytoplasm. mRNA is present, tRNA is present. Here, amino acid pool, inside amino acid pool, there are different amino acids. There are different amino acids like, we can say every amino acids, right? So, methionine is there, leucine is there, isoleucine is there, tryptophan is there. Okay, then we can say valine is there, alanine is there. Okay, every amino acid is present inside the amino acid pool. So we can say the first step is to transfer the amino acid to tRNA. Okay, what is the first step? We'll call it as activation of amino acid. Okay, activation of amino acid. That is the first step before translation steps. Okay, activation of amino acid. Why it has to get activation? It has to meet the energy requirements. Okay, it has to meet the energy requirements. So, in order to meet the energy requirements, this amino acid has to be connected with ATP. Okay, it has to connect with ATP by after getting activation. Okay, this will be connected. This activated amino acid will be connected to the tRNA molecule. Okay, it will be connected to the tRNA molecule which is having anticodons. So, if you are considering an example, methionine. So, we can say methionine. First, what is the step? Methionine. Methionine will be get activated with ATP molecule, and next this methionine will be connected to the 3' position of tRNA. Okay, let's see the steps in detail. See. First one, it is charging of tRNA. Okay, charging of tRNA. That means amino acid. tRNA, charging of tRNA means amino acid is directly given to the tRNA molecule. Okay, so see what happens. Formation of a peptide bond requires energy. So, if you are considering amino acids in after translation, this is the first amino acid connected. Then one next amino acid will be coming. The two amino acids are connected with the help of which bond? It is with peptide bond. Okay, so for the formation of peptide bond, what is required? Energy required. And for the energy requirement, amino acid will be connected with the ATP. See, for this amino acids are activated. Amino acids are activated with ATP. Okay, and then it is linked to their cognate tRNA in the presence of which enzyme? Amino-acyl tRNA synthetase. It is very important. Okay, activation of amino acid, charging of tRNA. It is done with the help of one particular enzyme. What is the name of that enzyme? It is amino-acyl tRNA synthetase. Okay, amino-acyl tRNA synthetase. So, we can say when this amino-acyl tRNA synthetase helps the amino acid to get activated with the ATP molecule, that process, or then we will call the tRNA as a charged molecule. The tRNA as charged molecule. Okay, got it? So, next, what is the step? Initiation. Now, initiation takes place. What is initiation? I told you before, AUG is there present in the mRNA. So, AUG will be acting as the initiator site. Okay, so what happens here? The RNA molecule will be attached. See, if this is mRNA, what happens? Ribosome, smaller subunit of ribosome will be first attached to the mRNA. Okay, then it will be searching for the start codon. It has to be started. The process has to be started, right? So, it will be searching for the start codon. Once start codon is found out, what happens? The second larger subunit will be attaching to that particular site. Okay, so see, mRNA binds to the small subunit of ribosome. See, this is the smaller subunit. Towards this subunit, what will be attached? mRNA will be attached. Okay, so what happens now? The larger subunits bind to the smaller subunit to complete the initiation complex. Okay, after finding the start site, if there is start site, what happens? The larger subunit will come and attach. Okay, then larger subunit has two binding sites for tRNA. I told you, if you are considering the structure of larger subunit, it has three different sites. Okay, what are the three different sites? It is E site, P site and A site. Okay, so E site is the exit site where the tRNA molecule is moving. Okay, moving out of the ribosome, tRNA molecule. Once tRNA has to enter with amino acid, the same tRNA has to get connected with the next amino acid. Then the tRNA which removes the amino acid has to move out. This process has to be done. What it has to be done? If it is a long mRNA, ribosome is attached. Ribosome now is attached. Okay. Here what happens? A tRNA molecule. There is a tRNA molecule. It has to enter. Then it has to move. Then what happens? It has to leave with the tRNA. Simple tRNA without amino acid. Okay. So you have to be thorough with the concept first. Then you have to look into this. Okay. Next what happens? Amino acid tRNA binding site is there. That is A site. And peptidyl site is there. That is P site. What is amino acid? First you have to think the first every time first amino acid will be coming to the P site. Later on this P site will be moving into A site. From A site again it will be moved to P site. Then to E site. tRNA will be moving to E site. The second amino acid will be coming to A site. It is connected with the next amino acid moving to the next. It happens. Okay. Then see, initiation codon is always AUG. What is the initiation codon? It is also mentioned that it is AUG. Methionine tRNA complex would have UAC as the anticodon site. Okay, if you are considering this as mRNA, this as mRNA, this as AUG. Our tRNA molecule which is having UAC. UAC will be coming and this site will be considered as P site. This site will be considered as P site. Towards this side, what is there? A site is there. Towards this side, E site is there. Okay. So next amino acid tRNA with amino acid will be coming to which site? A site. Okay. The second amino acid will be coming to which site? A site. Okay, once it comes, then this P site and A site is having two amino acids, right? So it will be connected. The first amino acid and the second amino acid will be connected with peptide bond. Later on, there is a free tRNA here. That free tRNA will be moving to the E site and the process continues. That process, the formation of peptide bond is known as elongation. Okay, see, I told you, P site, the first codon of mRNA binds with anticodon methionine tRNA complex. Always think that first codon is mRNA. Sorry, methionine. Okay, first codon, initiator codon is methionine. Okay, so how this methionine comes? Methionine comes in the which position? Methionine comes in the which position? Okay, methionine comes in the P site. Methionine comes in the P site. Okay, then what happens? Another amino acid tRNA complex with an appropriate amino acid enters the ribosome and attaches to the A site. So second step is there is first tRNA here, second tRNA is present here now. Okay, that is tRNA. Okay, first tRNA is present at the P site, second tRNA is present at the A site. Okay, after coming, see, two tRNA with two amino acid will be present. So this is the first amino acid, right? And here you have the second amino acid. Both these amino acid will be connected later on with a bond known as peptide bond. What bond it is? Peptide bond. Okay, then what happens? This amino acid, if this amino acid is connected with this, there is no amino acid in the P side. There is no more amino acid in the P site. Okay, so we can say this amino acid won't be there. Okay, so this two amino acid will be now connected. Two amino acid will be connected. Okay, there is no amino acid here, only the tRNA will be present. So this tRNA will be moving to which side? E side, that is exit side. Here now it is free. Okay, here now it is free. Here now two amino acid is connected with peptide bond. Later on what happens? This tRNA will be moving to, I told you tRNA will be moving to E side. There is no tRNA now in the P side. The step, okay? There is no tRNA here. Okay, there is no tRNA here. What happens? The second tRNA which carries the two amino acid. Here see, tRNA is present, two amino acid is present. So this tRNA will be, this tRNA, okay, so we can say we can say the tRNA which was present there. Okay, tRNA with two amino acids. Okay, with two amino acid is now present at the A site. A site is free now, right? A site is free. A site is completely free. Another amino acid comes here. Okay, another amino acid. The third amino acid, third tRNA will be coming. Okay, third tRNA will be coming here. So what happens here? Third tRNA with third amino acid is present now. Okay, so what happens after this? After reaching here, this tRNA molecule. See, this has two tRNA molecule, right? This tRNA, both the tRNA molecule. Okay, both the tRNA molecule will be transferred to this tRNA. Okay, so three tRNA will be, sorry, three amino acid will be attached together. Three amino acid will be attached. So this amino acid will be, this amino acid will be moving. Okay, so this amino acid will be moving to this side. So how many amino acid will be present? How many amino acid will be present? Three amino acid will be connected. Here only tRNA is present. Okay, what is present? Only tRNA is present. This tRNA will be moving to the next side, that is E side. Okay, now here how many amino acid is there? Three amino acid will be present, right? This process continues. Okay, first every time think first amino acid will be coming to this side, second to this side, then it got a connection. Okay, first to this side, second to this side, then it got a connection with the second one, then the second one leaves. Okay, the second one, the tRNA leaves, again a new tRNA comes and the process continues. Okay, that is happening here. Its anticodon binds to the codon between the mRNA and a peptide bond is formed between the first and second amino acid in presence of an enzyme known as peptidyl transferase. So, I told you amino acid, first amino acid will be transferred to the second amino acid and that amino acid transfer is done by another enzyme and this enzyme is known as peptidyl transferase. Okay, peptidyl transferase. Okay, finally, when this mRNA reaches into the terminator codon. Okay, what are the two different terminator codons? What are the two different terminator codons? Okay, before that, the process is known as the process of, okay, the process of uncharged tRNA moves from the P side to E side. Okay, there is no amino acid in the tRNA after the transfer of amino acid from P side to A side. Okay, so we can say that uncharged tRNA, okay, that uncharged tRNA will move from P side to which side? Exit side. And that process is known as translocation. This is important, okay? You will get a one word question from this. Okay, what is that? Translocation. Okay, and again what happens? I told you the process continues. Later on, what is the next process? Termination. See, if this is an mRNA, the process continues. Translation is continuing. Okay, the translation continues, and if it reaches to a site known as UGA. UGA is a terminator codon, right? This does not code for any amino acid. There is no amino acid for UGA. So, we can say the termination or the stop codon. If the stop codon is present in the mRNA, we can say the translation process will stop. Okay, translation process will stop. So, see this when amino acid tRNA reaches the termination codons like UAA, UAG and UGA, the termination of translation occurs. Okay, if it happens, what happens? If it reaches to the three codons, three different codon, what happens? UGA, UAA and UAG, UGA, UA and UAG. Okay, it is important. Okay, what is the first starting codon and what is the terminator codon? It is very important. Keep in mind and study well. Okay, then the polypeptide and tRNA are released from the ribosome. Once translation is finished, once translation is done, mRNA, this larger molecule will be detached, the smaller unit will be detached, the mRNA will be detached, and you will get many amino acids connected together. Okay, many amino acids connected together. See, amino acid will be connected together with peptide bond, and this particular molecule will be known as a polypeptide. What this is known as? It is known as a polypeptide. Okay, and this polypeptide is known as protein. Okay, this polypeptide is known as protein molecule. Once the process is done, what happens? Everything detaches. The larger subunit detaches, the smaller subunit detaches, mRNA detaches, and you will get an amino acid, sequence of amino acid, and that sequence of amino acid connected with the peptide bond is known as polypeptide, and this polypeptide is known as simple, it is protein. So from DNA you got mRNA, from mRNA you got protein. Got it right? Got it right? Okay, so this is the structure of process of translation which is given in your textbook. See, there is 3 different sides. Okay, and along with this is P side, this is E side, where this what we can say, where uncharged tRNA is moving out. Okay, uncharged tRNA is moving out, P side, then where the peptide bond is forming, then this is A side where a new tRNA is coming with the amino acid. Okay, so it is also important to note down what are the different things which is present inside this. See, there is an mRNA which is having a 5 prime, 3 prime end. There is a ribosome, there is a tRNA. Okay, tRNA molecule which is having an anticodon side and which is having a amino acid. Okay, got it? Got it? If you get it means you can give me a thumbs. Okay, so next we will be discussing with gene regulation. Okay, what is gene regulation? Every time, every genes are not necessary. Okay, some genes have to be regulated or their process have to be regulated. Okay, and in we can say in prokaryotes, this gene regulation can be done in an operon system. Okay, operon system. We can say, see, when we have studied transcription, we have understood the term cistron, right? Cistron, what is monocistronic condition and what is polycistronic condition. We can consider prokaryotes as polycistronic, right? Polycistronic. So how this gene can be regulated? How could be the gene can be regulated? It can be regulated in transcriptional way. Transcription. If transcription is stopped, okay, gene is regulated. There is, if transcription is stopped, what happens? Translation also what happens? Transcription. After transcription, mRNA synthesis will be taking place, right? So after mRNA only, after the synthesis of mRNA only, we can get the process of materials of translation steps. It can be started to initiate the steps of translation, right? So we can say if transcription is blocked, translation what happens? Okay, so next if you are considering translation, translational level gene regulation is present, and if you are considering that translation, if translation is blocked, what is the result? Protein synthesis is stopped. Okay, so in different methods we can regulate the genes, and we will be discussing here the lac operon. What we are discussing, it is very important with the exam point of view. So you have to study this lac operon. Okay, what is actually lac operon or lactose operon? First you have to study that it is an inducible operon. What operon it is? Inducible operon. Okay, inducible operon. Okay, inducer operon. Okay, so what does, okay, so see here you can find promoter region, then you can see the inhibitor genes, then you can find the promoter sequence. Okay, let's discuss the structure of lac operon. So see, this is the promoter site. I told you, we have already learned in the case of transcription, the transcription unit consists of the promoter site, the terminator site. In between there is structural genes, right? So when polymerase enzymes comes and binds to the promoter site, then when it moves, what happens? The genes for mRNA for the structural genes will be synthesized. Okay, the same thing is happening here. See what happens in the presence of lactose and in the absence of lactose. That is the question. Okay, so same idea, students. So we have to, in order to study this condition, we have to study the situations of lactopron. Okay, when does the lactose operon will be working? So you know that bacteria can survive in glucose, right? See, if you are looking, if you are considering this is as a Petri plate, this is a Petri plate. Okay, this is a Petri plate. Okay, I have added the medium as glucose. What I have added? I have added the medium as glucose. Okay, and then what I have done? I have striped this particular bacteria. For example, we can say E. coli. Okay, E. coli is a bacteria which I have allowed to grow. Okay, which I have allowed to grow. So, if I am allowing this bacteria to grow in the presence of glucose, what happens? It will take glucose as its nutrient, right? We need food for the nourishment and for the growth. The same thing is for whom for whom for the E. coli too or the bacteria too. Okay, for their growth and nourishment and development and reproduction, it requires the glucose. Okay, so glucose is finished. They have utilized all the glucose molecule which is present inside the plate. Okay, so what happens next? What is next process? What is the next step? So, if I'm adding lactose, okay, there is no glucose present. Glucose is completely utilized. Okay, then next I am providing, what I am providing there? I am providing lactose. Okay, I am providing lactose. If there is no glucose, if there is no glucose and if there is lactose, which operon will be working? Which operon will be working? Lac operon will start working. Okay, why lactopron should work? See, inside the lactopron, there are three different structural genes. Okay, three different structural genes are present. And what are the three different structural genes? It is Z, Y and A. What is the genes? It is Z, Y and Z gene is there, Y gene is there and A gene is there. Z gene will code for, Z gene will code for beta galactosidase. Okay, beta galactosidase. Permease is there. It will Y for permease and A for transacetylase. Here, beta galactosidase is very important. Beta galactosidase is very important. Galactosidase. Okay, it will code for permease. Okay, A gene for transacetylase. Okay, so beta galactosidase, see what is present? Lactopron will be functioning and inside lactopron, what is the genes? Structural genes are present, ZY gene. Among the Z gene, it is the gene which is coding for beta galactosidase and beta galactosidase have the ability, okay, beta galactosidase has the ability to break down lactose. Okay, lactose into galactose and glucose. Dear student, it is breaking the lactose molecule into galactose and glucose molecule. Okay, so here you can find glucose molecule. Again, the bacteria will get the glucose molecule and they will be utilizing the glucose for their requirement, for their nourishment and for their growth. Okay, so basically simple thing, lactose is given, lactose has to be broken down into its monomer units, that is galactose and glucose. Once glucose is present, glucose can be utilized by the bacteria. For that purpose, lactopron should be on. Okay, but at some time, if there is glucose, if there is enough glucose, the lac operon will be off. Okay, if there is enough glucose, lac operon will be off. When glucose is completely utilized, lac operon will be on. Lactose, if lactose is present, lac operon will be on. So we can say lac operon is an inducible operon. Remember the point, okay? Lac operon is an inducible operon. So let's see the structure of lac operon. See, if you are looking into the structure of lac operon, it has a promoter site. It has a site for inhibitor synthesis. Okay, then again it has a site for the structural gene, then it has a site for an operator region, then it has the region of structural genes. Okay, what happens here? If there is no inducer, that means if there is no lactose. Okay, if there is no lactose, what happens? This promoter is active. Okay, this promoter site is active. Polymerase will come and bind to the promoter site. It will produce the repressor mRNA. Okay, if once the repressor mRNA is produced, the repressor protein molecule will be produced after translation. Okay, what is produced? Repressor protein will be produced. Once repressor protein is produced, this repressor protein won't be sitting simply. They will go and bind to the operator site. Okay, once repressor protein is formed, it will be going and attached to the operator site. Okay, so when you can find, see, operator site, the inhibitor molecule or the repressor molecule is binding towards the operator site. So what happens if a promoter region, here you can find the promoter region, right? So when the polymerase enzymes comes and bind to the promoter region, the polymerase enzyme cannot move towards to the further position. Why? It is blocked. Okay, operator region is blocked with repressor molecule. Got it? So next what happens? Next, the complete utilization of glucose is done. Okay, the complete utilization of glucose is done. Then what happens? Lactose is added. Okay, lactose is added. If lactose is added to the medium, the permease molecule, okay, the permease will take the lactose, simple lactose molecule into the bacteria, inside the bacteria. Okay, it will be taking, permease will be taking a small amount of lactose into the bacteria. So once it is entering, the small amount of bacteria is, so small amount of lactose is entering into the bacteria, what happens? This lactose molecule, the small amount of lactose molecule will be acting as the inducer. Okay, it will be acting as the inducer molecule. So what is the speciality of this inducer molecule? Inside this, if inducer molecule is inside the bacterial cell, this inducer molecule go and bind to the repressor molecule which is present in the operator site. Okay, it is going and binding to the operator, it is going and binding to the repressor molecule which is present in operator site. It will be removing the repressor protein. So once it removes the repressor protein, see, this promoter site is free. Okay, promoter site is free, operator site is free. The polymerase enzyme which is binding towards this can move. Okay, it can move towards this side. So when it is polymerase enzyme is moving from this side to this Z gene. Okay, Z gene, what happens? Beta galactosidase will be produced. If beta galactose is produced, what happens? This beta galactosidase will helps to synthesize the glucose molecule from lactose. Okay, then again Z gene, then next gene is Y gene. Permease will be produced. Okay, permease means permease helps to take the lactose molecule into the bacteria. So next one, transacetylase. Since the date, the function of transacetylase is unknown. Okay, the scientists also don't know why transacetylase is produced. Okay, but the two enzymes, the other two structural genes, it has the function. First one, it is beta galactosidase. It converts into glucose. Then second one, it is permease molecule. And what is the function of permease? Permease takes the lactose into the bacterial cell. Okay, so you understand the function and why does the lactose operon functions? You got it, right? So that is very simple. My dear students, lactose operon is a sure question. Okay, it is a very sure question for if you are going with a bio background. If you are going with a bio background, for sure, I am saying you have to again and again study the same thing. Okay, so if your base is very much clear now, you can move ahead. Okay, so next you have to study with tryptophan operon. Then you have many different types of operon to study. Okay, not only this lack operon, but it is very interesting. Okay, see, something is coming and binding, something is taking over this some place, and it is making a block, so the polymerase enzyme cannot go. Then again, what happens? This another molecule, the inducible molecule is coming. It is taking this from operator, repressor molecule from the operator side. Then the operator is free, promoter is free, binding, polymerase enzyme comes and bind, then it moves. Okay, so it is very simple and logical. If you apply some logics, you can get it easily. Okay, so you are thorough, right? It is very important. You have to study this. Next, the last and final topic of this chapter, we will be discussing with human genome project. Okay, if you're looking into human genome project, what is a genome? The entire DNA in the haploid set of chromosomes. See, in the human beings, we have two sets of chromosomes, right? Diploid sets of chromosomes. So we can say if the chromosome which contains the haploid, the entire DNA in haploid chromosome, then we will call it as genome. Okay, what is actually human genome project? Inside human genome project, we can say the sequences present in the human genome was identified and it was studied. Okay, so we can say we have to study what are the goals of HGP. It is very important. Goals of HGP, Human Genome Project. It is to identify all the estimated genes in human DNA. What are the different genes present in human DNA? It has to be studied. That was the main goal of HGP. Okay, so next goal to determine the sequences of 3 billion chemical base pairs that make up human DNA. See, if you are considering human DNA, it has 3 into 10 raised to 9 base pairs. Okay, 3 into 10 raised to 9 base pairs it has. So, to determine what are the different sequences present in this 3 billion chemical base pairs, what happens? Human Genome Project was established. Okay, then the third one, to store this information in database. Okay, once it is identified, it has to be stored into our database. That is also one goal. Then D, to improve tools for data analysis. Okay, once the data is studied or once the data is taken, the data has to be analyzed with different tools and technologies. Okay, it has to be modified, and that is also one of the goal of HGP. Then, to transfer related technologies developed during the project of society to other sectors of society. That means, see, if human genome project is done with the help of bioinformatics too. Okay, not only biology, it is connected with bioinformatics. So, the technologies has been merged and it is used for the well-being of human beings. Okay, so that is the goals of HGP. Next, you have to study the salient features of Human Genome Project. What are the different salient features of Human Genome Project? If you are looking at the salient genome project, salient features, see, I have told you that there are 3 billion. Okay, 3 billion base pairs. In particularly, we can say 31,064.7 million nucleotide base pair was there. So, once the Human Genome Project was done, we identified the what are the different genome or what are the different base pairs which is present inside the millions or which is present inside the complete or entire DNA of human being. Okay, then so total number of genes present. See, the goal was also to identify the genes, not only the base pairs, it was also to identify the genes. So we can say the total number of genes was about equal to 25,000. Okay, how many genes you understood? 25,000 genes was present in the case of human gene or human genome. See, what is the difference between gene and the chromosome, or what is gene actually? The portion of DNA, right, which gives the characteristic phenotype to our organism. So see, We can consider the hair color. It is present. The hair color is present as one of the gene in the chromosome. Okay, eye color, it is also present as one of the gene in the chromosome. So skin color, it is present as the one of the gene in the chromosome. If there is any alteration happens, everything will be altered. Okay, so 25,000 of genes was identified and it was studied. Okay, then see average gene of 3,000 bases. Average gene, if you're considering one particular gene, one gene contains around 3,000 bases. Okay, how many bases? 3,000 bases it was. Okay, and the size varies. The largest non-human gene is dystrophin on X chromosome, which contain 2.4 million base pair. Okay, then next one, 99.9% of nucleotide bases are identical in people. Okay, they are identical. 99.9% of human genome was identical. That means I and you have almost same type of DNA, but 1% has difference. It has 1% difference. That not 0.1 percentage. That 0.1 percentage differences of DNA make you a unique particular person. Okay, so see the speciality of that 0.1 percentage, right? I have a different smile, you have a different smile. I have a different face, you have a different face. I have a different character, you have a different character. Everything comes from genetics. Okay, so we can say that 0.1 percentage makes us unique in nature. Okay, the next one, 50 or 50 percentage of discovered genes are in all chromosome I. Okay, all in different chromosomes, we can say. Then next one, chromosome I has the most gene, that is 2,600, 2,968 genes, and Y has the fewest gene. Y chromosome is present in the males, right? In males for the sex determination, Y chromosome is very important. So the Y chromosome will be giving how much genes? It has 231 genes. Okay, and chromosome 1 has 2,968 genes. Okay, and we can say less proteins have been identified from the human genome. Then repeated sequences make up a very large portion of the human genome. See, repeated sequences are present. Okay, so this repeated sequences are stretches of DNA sequences that many times they have no direct and repeated coding functions, but they shed light on chromosome structures, dynamics, and evolution. Okay, it has the function in different areas like structure, dynamics, and evolution. Okay, understood? Got it? So that is all about the Human Genome Project. Okay. Okay, Human Genome Project, you have to study about what is Human Genome Project, then what is the goals of Human Genome Project, and what are the silent features of Human Genome Project. Okay, next coming, see Rice Genome Project. Rice is one of the major thing that we are consuming every day. Okay, the need of the population increases, the need of food is also increased in the human beings. Okay, so what happens? The requirement of rice cultivation also increases. So we have to understand what are the different genes present in the rice, so we can improve the quality and quantity of genes with the different technologies like biotechnology. Okay, so let's see, rice is one of the most largely consumed food. Okay, it is most consumed food. It is has when population increases, the requirement for rice also increases. So rice genome project has been launched to increase the production of rice with different technologies. I told you, with we can say biotechnology. See, there are two different chapters in our textbook with dealing with biotechnology, its principles and its application. There we have studied how the genes will be taken and it is cutted and it will get a cut and it will be pasted to the different organisms. Okay, so with the same technology we can say rice genome project has also been done. Why rice has smallest genome of 430 MB nucleotides located on chromosome 12. Okay, it has on chromosome 12 it has 430 MB of nucleotides. Okay, then next who has taken or who has initiated this project. It is a joint project of National Institute of Aerobiological Sciences, Forestry and Fisheries, then Ministry of Agriculture, Forestry and Fisheries, Society for Techno Innovation of Agriculture Genome Research Program. Okay, everyone comes together and they have done which project? Rice Genome Project. Okay, and which is the plant which was most taken? Arabidopsis was the plant. It is an experimental plant for rice genome project because it has a fast life cycle and can be easily grown. These are the two conditions why I have chose Arabidopsis for the experiment. Okay. Then it has smaller genome and high diversity and helps in the enhancing the molecular project. Why Arabidopsis? It has a smaller genome, very small genome in present, and it has a high diversity and helps in the enhancing of the molecular products. Okay, so need for sequencing rice genome. Why rice genome sequence? Okay, why rice genome sequence? To know the functioning of genes by accurate gene sequencing. To know the function, see, I told you, it is to improve the quality and quantity of the rice products. See, why rice has to be, why the proportion of rice cultivation has to be increased. As population increases, the need for rice also increases, right? So, if the technology is integrated with agriculture, then more production will be taking place. Okay. So, in order to have the more production, if the particular gene is increased or improved, we'll get the production as fast as possible, like in BT cotton, BT brinjal, etc. Okay. So, we can say it is to know the functioning of genes by accurate gene sequencing. It is important for agronomic tastes which requires mapping of genomic sequences. Then improvement of other cereals also will become easier if the genomic sequences is done. Okay, so that's all about rice genome project, human genome project. Okay, clear? Understood, right? So this is the end of the chapter. So today we have studied with translation, right? Translation, what are the requirements for translation or translation machineries? What is simply we can say it as mRNA, ribosome, amino acid, and then tRNA. What are the different steps? At first, we have to activate the tRNA molecule. Okay, charging of tRNA with activation of amino acid with ATP molecule. This ATP, why ATP molecule has to be attached with amino acid? It is for the requirement of peptide bond formation. Two different amino acids get connected with peptide bond. If the peptide bond has to be done, what is required? ATP molecule is required. So first process is charging of tRNA. Second process is initiation. Initiation is or can be only done with the one amino acid, that is methionine. AUG is the codon. Okay, so there are three different sites in ribosome, that is EPA site. First amino acid will coming and binding to the which side? P side. The second amino acid with tRNA will be coming to the second A side. Then later on from P side, the amino acid will be moving to A side. Okay, then it will be again coming into P side, then it will be removed in from the E side. That process will be going on. This we can say the process of movement of tRNA, uncharged tRNA from P side to E side is known as translocation. So different enzymes has been taking place in different steps. You have to study that. When it comes to the tRNA with when the mRNA is having the stop codon as UAA, UGA and UAG, we can say terminations takes place. Okay. Then we have discussed about the operon system, lac operon, right? How gene can be regulated. Gene can be regulated with different technologies or techniques. That is with the help of, we can say, transcription method is there, translation is there. Then we can say that transcriptional and translation method will be there, right? Then later on we have discussed with the last portion, that is human genome project and rice genome project. Okay, it is very important that I have mentioned as very important as lacopron and translational machineries. In human genome project you have to study with what are the goals, what are the salient features. In rice genome project, what is the necessary, why it is necessary, and also what are the different what we can say, what are the different communities that come together for rice genome project. Okay, why rice genome project has to be done. Okay, that's all about this chapter. this chapter is finished. We'll be seeing in next class with another chapter. If you have any suggestion, you can comment in the comment box, okay, which chapter that has to be gone for the next live. Okay, so that's all about today. Thank you so much. On next Saturday, our Nandini Miss will be coming with electromagnetic induction as the chapter, and it will be the part 2, second part of the chapter. So stay tuned until Saturday. Thank you.